I'm introducing here Megan Lee. She is the publisher of the first author of the paper that sequenced more than 1,000 ancient genomes, and she's studying the coevolution of pathogens in humans and the natural selection that imprinted different characteristics and genotype in Europeans' genomes. So thank you, Megan, and she's from the University of Texas, Austin.
Can you all see my screen? Great. Okay, thank you guys so much for having me here today. I'm really excited to share with you all my undergraduate work on detecting natural selection using ancient DNA. So ancient DNA is a powerful technology that allows us to sequence genetic information from skeletal remains that are thousands of years old. This has revolutionized the study of human population history, but it's also powerful for looking at genomic evolution over time because it lets us directly track changes in allele frequency between generations. Previously, this was not possible due to limited sample sizes, but now we've moved from looking at singular ancient genomes to sequencing thousands of samples available across both time and space. The vast majority of the data comes from Europe and is limited to the past 10,000 years. So studying this period and geographical region is interesting because it can give us insight into the processes of natural selection that acted over 10 millennia of profound change in human lifestyle. So these include the transition from hunting and gathering to farming, which resulted in major changes in diet, as well as increased population density and closer proximity to animals. This period also includes a transition to state-level societies, which was facilitated by metalworking and led to large population densities and long-distance exchange of goods. And then additionally, there have been several ancient DNA studies that also sequence bacterial and viral pathogens that cause epidemics in the last few millennia, and these include smallpox, the Black Death, and tuberculosis. So all of these events could have potentially induced selective pressures on the populations living at that time. So by studying ancient DNA in a time transect, we can gain insight into the genomic evolution that occurred in tandem with these cultural transformations. And it turns out that in Europe, these cultural transitions have been accompanied by major demographic transitions as well. So these have been revealed by multiple studies that actually use ancient DNA to identify gene flow, human migration, and mixture. So in total, they report thousands of ancient samples, and these many, many studies provide excellent fine-scale models for the demographic history of Europe. So they can be summarized by three major events. First, we have the movement of people from Anatolia, who brought farming technology into Europe and mixed with local hunter-gatherers. Then second, we have the arrival of pastoralists from the Pontic-Caspian steppe during the Bronze Age, which resulted in mixture with European farmers. And then third, in the historical period, we see homogenization of these three different ancestry types: the European hunter-gatherer, Anatolian farmer, and steppe pastoralist. And it turns out that rather than being a confounder, population mixture can actually be used to detect natural selection by finding allele frequency changes that vary more than expectation. And this is an approach that was first described in Matheson 2015. So suppose we take a look at that first epoch that I described, the Neolithic. So the first farming populations of Europe are a mixture between the Anatolian farmers and European hunter-gatherers in an 85 to 15% ratio of overall ancestry. So these mixing proportions are a genome-wide estimate, but now suppose we pick a single mutation, and in hunter-gatherers, this allele had a 50% frequency, while in Anatolian farmers, it had a 10% frequency. We can then compute the expected allele frequency of the admixed population by taking a simple weighted average of the allele frequencies and the mixing proportions. So in this case, 15% times 50% plus 85% times 10% gives us an expected allele frequency of 16%. However, we can also look at the European farmers several generations after that initial mixture and look at what the allele frequency is then. So, in this example, let's suppose that's 70%. So if that observed allele frequency significantly deviates from the expected allele frequency, then this is consistent with natural selection. So we quantify that deviation using the likelihood ratio test, which is a goodness of fit test that compares the maximum likelihood estimate of the observed allele frequency to a null model based on the genome-wide mixing proportions. So we apply this method genome-wide, looking for variants that deviate significantly from their expected allele frequencies. Then, as I mentioned earlier, Matheson 2015 used this approach in a scan with modern individuals as their admix population. But thanks to increased ancient DNA sample sizes, we can now apply this method in slices of time. So we did this across 3 time periods, which reflect the 3 major cultural transformations. So the transition from hunting and gathering to farming, the Bronze Age, and the historical period, each one representing about 3,000 years post-admixture. And what's really unique about taking this time stratified approach is that by dividing the past 10,000 years into these 3 periods, we're able to identify the signals with better temporal resolution and look at them in relation to the cultural changes that occurred during these times. So these figures show real results from our scan, and they're a cool visualization because it shows the changes in allele frequency over time for a single SNP. So the populations are arranged by time period, so the more recent populations are toward the right, and the dots show the maximum likelihood estimates of the observed allele frequencies for each population. And then those three populations with the colored bars are our admix populations. So the dashed lines show us what we expect the allele frequencies to be. So panel A is a SNP that is in very high LD with CCR5 delta 32, which is something that provides protection against HIV in European individuals when they have the homozygous form. So this was previously reported to be under selection, but from our results, we don't see strong deviation from expectation in any of the epochs. And then on the other hand, if you look at LCT in panel B, that's the lactase persistence allele. So this is a classic signal of selection that frequently appears in these selection scans. And you can see that LCT doesn't get introduced into Europe until the Bronze Age. But then once you hit the more recent historical period, the allele frequency just shoots up dramatically. So now let's get into our full results for each period. So this is a Manhattan plot after we performed correction for genomic inflation. So we essentially corrected our statistics, so the median chi-squared statistic was equal to the median from the null chi-squared distribution. Then you can also see on here that there are some SNPs that reach significance on the plot, but they're grayed out. And this is because we also applied an additional filter of only keeping sites for which there were at least 3 significant SNPs within a 1 megabase window. And that would help us filter out through noise caused by genetic drift or bad genotypes. So this plot is for our earliest period, which is the Neolithic, and reflects the transition from hunting and gathering to farming. And as one might expect from a sudden transition to a primarily starch-based diet, we see a number of loci associated with things like insulin secretion, diet, and body weight. So for example, we see a signal of selection on FTO-IRX3, which is a major obesity susceptibility locus, but the variant that we find actually decreases expression of IRX3. We also see BDNF, which is associated with regulation of body weight. And then there are various loci associated with insulin secretion and circulating glucose levels. So we have PTPRV, ENSA, and MAF. And the Neolithic is also a time when humans began to live in close proximity to livestock. And we observe a number of immune-related signals, perhaps a result of humans being exposed to zoonotic pathogens. So we see HLA, which plays an important role in the human adaptive immune response. There's also FUT2, which determines the secretion status of the ABO blood group antigens. And then something else that's interesting is that our significant FUT2 variants are associated with plasma B12 levels, and that's a vitamin that's largely unavailable from plant-based food sources, but it's plentiful in animal products. Then some of the other signals that we find are IL-1R2, which is expressed on lymphoid and myeloid cells and has been implicated as a susceptibility locus for some autoimmune diseases. In addition, we find CACMB1, which is a regulator of T cell function, and PPIL2, which is related to resistance from HIV infection. And then our last signal that we see is FAM49B. And this signal is interesting because FAM49B has been shown to confer resistance to salmonella infection, and there has been previous evidence dating salmonella enterica's adaptation to human hosts to the Neolithic. So this could be a possible cool example of human pathogen coevolution. So in total, we found 11 signals of selection in the Neolithic, but we wanted to also compare our results to the results from the Matheson 2015 paper, which again applied the same method, but used modern individuals instead of ancient individuals as their admixed population. And then we find that the only overlap between our Neolithic results with the Matheson 2015 paper is HLA, which is not pictured in this Venn diagram. So then if we jump forward to our second period, which is the Bronze Age, we see a couple of pigmentation-related genes. So we see a signal of selection on SLC45A2, which is an allele that plays a role in light skin pigmentation in Europeans, and has also been previously reported as a signal of selection in other studies. We also see OCAT ATRC2, which is an allele for light eye color. And then we again see signals on 5 immune-related genes, including HLA. This could be perhaps due to the introduction of new pathogens with incoming steppe pastoralists, as well as introduction of horses as a new domesticated animal. And then we additionally find a couple of cardiovascular related genes. So AGT causes vasoconstriction and increases blood pressure, and ABCG1 controls tissue lipid levels. Then our last signal is HYDIN, which is related to sperm motility and male infertility. That we do caveat that there's a partial duplication of that region on chromosome 1 that's not annotated in HD19. So that could potentially cause a false positive. And then when we go back to looking at the overlap with the other scans, we see that the only overlap with the Neolithic is HLA, but both of the pigmentation-related genes were actually previously reported in Matheson 2015. But besides that, 7 out of 10 of our loci that we find are novel. And then finally, in the historical period, which is our most recent period, we see continued signals of selection on immunity and light skin pigmentation. So we still see HLA and SLC45A2, but our strongest signal is LCT, which, as I mentioned earlier, confers lactase persistence. And in addition to seeing LCT, we also see a lot of signals related to vitamin D. So for example, light skin pigmentation promotes vitamin D synthesis from sunlight. We also find SH2B3, which has been fine mapped to vitamin D binding. DHCR7 regulates the availability of 7-dehydrocholesterol for conversion to vitamin D. And 7-dehydrocholesterol is something that's very abundant in milk. So we think that the selection on LCT that we see here might be part of an oligogenic response to needing increased vitamin D levels. And then besides finding another immune-related loci in the toll-like receptor TLR, we also find FADs 1 and 2, which are involved in fatty acid metabolism. And then returning once more to our Venn diagram, we actually find that all of our historical period results have been previously reported in Matheson 2015. So in other words, we find that as our epochs grow closer to modern time, we report increasing overlap with the results from the scan using modern genomes. So this Venn diagram really shows the value of performing such selection scans in time intervals to uncover ancient selective signals that might later be masked due to subsequent admixture or drift. But then before I move on from talking about our monogenic results, I wanted to note that while our ability to time stratify our data increased to the selection scans' robustness and resolution, we attribute most of the improvement to the increased quality and quantity of available ancient DNA data. So we did an analysis checking how our power to detect loci was affected by smaller sample sizes. And we find that we recapture most of our signals with about 90% of our original sample size. But we don't see a clear plateau in the graph, which suggests that increasing sample sizes further is likely to continue to improve the power to detect new loci. And then to complement our picture of monogenic selection in the last 10,000 years, we wanted to test for polygenic selection on complex traits. So we did so by integrating our signal of allele frequency deviation with trait associations from genome-wide association studies, or GWAS. And we adopted a test from COIN 2021 that looked at polygenic selection in Oceania, where they used a haplotype-based statistic. So we modified that and instead created what we call a polarized statistic. So this takes the magnitude of our chi-squared statistic from our monogenic scan, and it's positive when there's selection on the trait increasing allele and negative for the trait decreasing allele. However, GWAS-based estimates can be poor for individuals and populations that are different from the groups in which the GWAS was carried out, perhaps due to ancestral or environmental differences. So one major problem that can lead to systematic biases is uncorrected population stratification in a GWAS, which is when axes of ancestry correlate with the trait that's being tested. So we took two measures that are expected to reduce statistical power, but increase robustness to population stratification. So first, for our primary analysis, we used GWAS summary statistics from the Biobank Japan rather than from the UK Biobank. So since West Eurasians and East Eurasians largely split from a common ancestral population more than about 40,000 years ago, the population structure in the Japanese data set is expected to be uncorrelated with the main axes of population structure among Europeans. So this is an idea from Chen 2020, which used Biobank Japan to look for evidence of selection on height in Sardinians. Then second, we limit our analysis to genome-wide significant associations, and we only considered effect directions from the GWAS statistics instead of effect sizes. So this is because when we look at significant loci that's shared between Biobank Japan and the UK Biobank, we found that over 95% of loci have the same effect direction, but effect sizes were only about 70% correlated. Then additionally, there has been evidence from model organisms, particularly plants, that suggest that across a wide range of traits, while the effect sizes of QTLs vary, effect directions are almost entirely conserved. So to test for evidence of polygenic selection, we first took the GWAS significant SNPs and computed their average polarized statistic. So on that slide, that average polarized statistic is represented by those vertical red lines. Then what we did is we drew an empirical null distribution from matched control SNPs. So we then calculated the average statistic for those matched sets, which on these slides is shown by those blue curves. So what we did is we looked for traits where the average statistic for genome-wide significant SNPs fell on the bottom 2.5 or top 97.5 percentiles of the null distribution. And the intuition here is that when we see that average fall, for example, on the far left of the distribution, then that's saying that when we look at the SNP significantly associated with the trait, we see that they have significant shifts in allele frequency in a trait decreasing direction compared to random SNPs of similar profiles. But before we jump into looking some of our results, we should also note that there are some caveats to using this approach. So, we do use these GWAS estimates from a modern Japanese population, but we're applying them to ancient Europeans who are very different in terms of things like their environment. And so while we do take measures in terms of, only using effect direction, there are caveats in terms of portability. So these results shouldn't be considered in isolation, and we can't definitively say whether selection has occurred or not, but rather we say that the significant results we see suggest that there's some evidence for selection favoring alleles in a trait increasing or trait decreasing manner. Yeah, so I wanted to highlight a couple of interesting things that we see in our results. So here, a shaded red box indicates evidence for selection favoring traits increasing alleles, while a blue box corresponds to trait decreasing alleles. So we tested 215 traits in total, and we see 12 significant results in the Neolithic, 9 significant results in the Bronze Age, and 31 significant results in the historical period. So firstly, in the Neolithic, we see evidence for selection favoring alleles that decrease body weight and BMI. And this is something that agrees with our monogenic scan results, such as the decreased expression of IRX3. And it's also something that's supported by archaeological evidence. But then second, most of our selection results are seen in the historical period, though a lot of these traits are likely correlated. We see a lot of selection on increased risk for cardiovascular disease and autoimmune diseases. Then, as I mentioned before, we created our primary selection statistic using the direction of effect rather than the effect size. However, we also ran our analysis using effect size in the selection statistic, and we find that our signals are broadly conserved. We also repeated our analysis using a within-sibling consortium GWAS, which the within-sib GWAS looks at genetic variation within families to better control for environmental and indirect genetic effects. So the trade-off is that there is vastly reduced power using the within-sib GWAS. So if you look at that fifth column in the table, it shows the number of within-sib SNPs in parentheses compared to the Biobank Japan number, and we see a sizable reduction. But looking at the results for the traits that we did have data for, we find some similar trends in our results. And then, as I mentioned before, we see evidence for selection favoring alleles that contribute to increased risk for cardiovascular disease. So one possible hypothesis for this could be because as humans started living in higher population densities with more mobility, they were subject to a greater infectious load, which would have increased selective pressure on inflammatory response, which secretes adipokines that underlie cardiometabolic diseases. So in summary, we found signals of selection on 25 genetic loci across our 3 time periods. All 3 epochs had immunity-related results. In the Neolithic, we also find diet-related genes. In the Bronze Age, we find pigmentation-related genes. And then in the historical period, we find many vitamin D-related genes. Then as our time periods grow closer to modern time, they also had increasing overlap with results from a scan on modern individuals. And then finally, our test on complex traits found evidence for selection favoring alleles that decrease body weight in the Neolithic and alleles that increase risk for cardiovascular disease in the historical period. So if you're interested in reading more about our project, we also have a preprint that's available on bioRxiv. So thank you all for your time. I also want to thank all of my collaborators and co-advisors for their discussions and support, and I'm happy to take any questions now.
Great. Thank you so much, Megan. That was really interesting. So just remember that the audience can now write that type their questions in the Q&A box, and I can read them out loud. And just to start with, so you are mentioning that there's still a lot of potential that if we keep on increasing the data, we can discover more and more of this genetic variation. And I was wondering, what are the current limitations nowadays? Is this a limitation of the technology? Is that there are no enough samples available? What do you think about that?
Yeah, so it's a limitation of just the number of available samples that we have. But there has been a recent increase in the samples that are being sequenced, both in terms of the time and also geographic regions. So that's really exciting.
Right, and this was just limited to Europeans. So I guess that there are other populations and regions that this is even more a problem, right?
Right, yeah, most of the data is from Europe. I think mostly because that's where people have been looking. But it would be exciting to maybe apply this method to other regions as the quantity of available data grows.
Mm-hmm. Thank you. So I have another question. So in your analysis, do you somehow take into account that maybe different genomes or genes may have different mutation rate, for example? How can you take this into account?
Yeah, that's a great question. So our our likelihood ratio test is comparing that. It's comparing our maximum likelihood estimate of the observed allele frequency to a genome-wide null based on mixing proportions. And that model is not as well, that might not work as well, as you mentioned, for some regions of the genome. For example, if you look at HLA, there are very atypical mutation rates. or rates of recombination. So it might not work as well for those parts of the genome, but very broadly, our model captures. We find that our model very broadly can apply to the genome. Yeah.
Great, thank you. And I may ask one last question. That is, so these studies, I understand that they help us to discover some historical facts that are unknown until now. And do you have any example of those? And I'm thinking, by instance, for example, some infections or some specifics on the diet that were not known previously and for which we don't have evidence. Do you have any examples of those?
So I think one of the cool examples might be, like the FAM49B signal that we found, which provides resistance to salmonella. And then, as we mentioned, there were studies that found that salmonella adapted to human hosts during the Neolithic. So that might be a cool example of like human pathogen coevolution.
Right, thank you so much, Megan. So I think we can, if there are no more questions in the Q&A box, so I think we can wrap it up here. And thank you so much again, Megan, for joining us. Thank you, Sophia. And thank you everyone for attending today's seminar. And we will be back next year, I think on the 10th of January, we have our next seminar series. So we hope to see you again there. Thank you, bye.